Welcome back, fellow scientists. So, yesterday we learned about karyotypes and we learned about how we make a karyotype and we learned a little bit about why we make a karyotype. So, this slide right here is the most important slide from yesterday. So, you remember that we have 44 autosomes, right? And we have two sex chromosomes, and the autosomes are identified by a number. So, the longest chromosome, the biggest chromosome in the human genome is 1, and then the smallest is 22, right? And then we have our sex chromosomes. And then we also came up with a shorthand way of summarizing the information contained in a karyotype. So we would start off with the number of chromosomes. So typical human karyotype has 46 chromosomes. And then we would list the sex chromosomes. So this would be X and Y. And then we would list anything else that might be different or might be abnormal with that karyotype. So that's a little review of the most important things. So now let's get on. So of course we know about determining gender, right? XX is female, XY is male. And then we ended with identifying some genetic disorders. So we're going to talk about those today. But before we talk about those, we need to talk about how we get these chromosomal abnormalities or how we get these chromosomal mutations. So and go ahead and write this down. This is part of your notes. So most chromosomal abnormalities or disorders are caused by non-disjunction. So we're going to define that. If you need to pause it, feel free. But I'm just going to keep on going because I want to keep this video as short as possible. So homologous chromosomes, we know that from meiosis one, right? The tetrads, right, of the butterfly, the two butterfly-shaped chromosomes. Homologous chromosomes that form tetrads do not separate during meiosis one. And if you look at the word, you can even kind of figure that out from the word. So junction, right? If there's a junction in two roads, there's a meeting of two roads. So junction means to stick together or to meet, right? To join. You can almost kind of hear that word, right? Disjunction obviously then is separating, right? And then if you say non-disjunction, that means that it's not separating, so that they're staying joined together. So as eloquent as that may be, let's go ahead and let's take a look at what this looks like. So go ahead, do your best to kind of draw this out. We're using our same color scheme that we did before, with two blue chromosomes from dad, two red chromosomes from mom. And so this big chromosome right here, this is what normally happens, right? So this is meiosis one, right? And then this is meiosis two, right? And so this big chromosome that forms a tetrad and then separates, right, in anaphase one, and then lines up, right? They don't show every single phase, but this is the basic idea. Lines up and then separates in anaphase two, and then we have one red chromosome per gamete over here. That's fully functional and it's made, right? This these chromosomes right here, those are going through non-disjunction. So this right here, that is the key point, right, in non-disjunction. The tetrads fail to separate for whatever reason, and so then what that does is that leaves two gametes with an extra chromosome, right, that they shouldn't have, because now they have two of the same chromosome, right? And it leaves two gametes that are missing a chromosome. So each of these gametes should have one of these little chromosomes, but they don't. Right? So they're missing a chromosome. So that's what non-disjunction looks like. Make sure that you have that written down. Make sure that you can kind of identify that and draw that and figure that out. All right, so we have a couple results of non-disjunction. We're going to deal with the autosomes first. Okay, so if a person has an extra chromosome 21, they have Down syndrome, and we'll go through these more in detail. This is just kind of like a pre-organizer for what we're going to talk about. If a person has a partial deletion of chromosome number 5, they have a condition called Cru de Chat. So that's not full non-disjunction. That's like just one teeny, 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 tiny little piece, right, sticking to this one, and then the rest of these things going on. And so then this little gamete right here would have a blue chromosome that's missing just a teeny, 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 tiny little end right there. And so that's, like we said, that's a condition called Cru de Chat. We'll get into this in a little bit. So those are the abnormalities that deal with the autosomes. And then abnormalities that deal with the sex chromosomes. We have three of them that we're going to talk about. There's more, and we get into that in AP biology a little bit. So there's Turner syndrome, right? And there's Klinefelter syndrome, and then there's trisomy X. And we'll talk about all these. You don't need to write these down right now. Sorry, apologize to those of you that already have. So Down syndrome, here is what a karyotype looks like for a person with Down syndrome, right? So notice that we have, first of all, two X chromosomes, which means that this person is a, yep, perfect female. And then you'll notice over here, we have all of these pairs of chromosomes except for chromosome 21, 1, 2, 3, we have three chromosomes. So the way that we would summarize this, the information contained in this karyotype, right, is 47, and not 46, 47 because we have an extra chromosome, right, 47, and then XX, right, so we'd still list the sex chromosomes female, and then that doesn't tell us why we have an extra chromosome, so then we need to say, okay, plus 21, so that means that we have an extra chromosome 21. All right. So when I was going through the education program at Whitworth, I had the opportunity to work with a student who had Down syndrome, and like he was amazing. He was really cool. He thought that I was perfect, that I did no wrong, and he thought everyone was perfect, did no wrong, and just like a really really cool person, really really cool guy, and just has a great outlook on life. I'm not sure if everyone who has Down syndrome is that way, but I've heard from a lot of people that they're just the kindest, sweetest, gentlest, most caring people in the world. So that's how they are, like temperamentally, like attitude wise. Physically, taken as a group, right? Most of them have small ears, small mouth, um, upward slanting eyes for whatever reason, mild to severe mental handicaps, right? Early intervention is key in the mild to severe. So we talked about genetics, how the genes provide a blueprint, but the environment affects it, right? So if you identify that one of your kids maybe has Down syndrome, then there are some early interventions to kind of train their brain that can help with that. Um, so flat nose, single crease in the palm of their hand, which is this is really weird, right? I know that all of you right now are doing one of these, like, oh, do I have a single crease? So just because you have a single crease in the palm of your hand, if you do, doesn't mean that you have Down syndrome. But kind of taken as a group, like there are a lot of people who have Down syndrome that have a single crease in the palm of their hand. Right, so that's Down syndrome, and then partial deletion of chromosome number 5, Cruda shot. So this is kind of hard to tell. So I'm gonna give the symptoms first, and then we'll do the karyotype. Oh, and by the way, you don't need to write down like all the things. I'm not going to ask you, okay, if someone has a single crease in the palm of their hand, what genetic disorder do they have? How this might work on a test is I'll give you a karyotype, and then you just have to tell me the name of the disorder or the syndrome or whatnot. You don't need to memorize all of these. All right, that's sorry, I should have said that before. So Cruda shot, right? Interesting to note that it's named, Crudeshat is French for cry the cat. So it's named because these people, as infants, have a really, really abnormal cry. It almost sounds like a cat, like meowing or crying, like really, really angsty. So that's why it's called Crudeshat, right? Microcephaly, so small head. Facial abnormalities, cognitive difficulties throughout life. Unfortunately, there's no treatment. And then the karyotype, I made this its own slide because you got to look really, really carefully. So partial deletion of chromosome number 5. You see this black band right here, and you see how that's not right here. Well, that small little tiny, teeny, tiny, minuscule missing piece of DNA leads to all of those difficulties, all of those abnormalities right there. So the way that we would write this individual has 46 chromosomes, right? And this individual is again a female, right? XX. But then there's something different about this karyotype. So it would be a minus 5P because there's a P arm and a Q arm, which is way beyond even AP bio stuff. But that's how we would summarize the information contained in this karyotype in CRUDA shot. All right, so now that we've dealt with the autosomes, now we're going to go ahead and deal with the sex chromosomes. So Turner syndrome, this is what a karyotype looks like for someone who has Turner syndrome. So you'll notice all the autosomes are normal, right? But then right here, our sex chromosomes, we just have one X and we have no Y. So the way that we would summarize the information contained in this karyotype would be 45, right? Because we're missing a whole entire chromosome. 45X. And that right there tells us why we only have 45. Some people add like a x0, right? Some people add like a x dash or whatever. Some people it's just 45x on the test. I'll accept 45x. That's totally fine. But that's how we would summarize that. So then, right, what does this lead to? What are kind of the symptoms? So I'm gonna try to be as delicate as possible, and you guys need to, like step up your game, be mature. So people with Turner syndrome have the physical appearance of a female. So you're babysitting, you're caring for an infant who has Turner syndrome. You got to go change their diaper, female, right? You guys understand what I'm saying? So in this, then as they grow older, then because the X chromosome kind of controls hormones and development and different things like that, there is a lack of primary, right? So those are organs and structures that deal specifically with reproduction, right? And secondary sexual development. So secondary sexual development would be ways that we typically distinguish between males and females, but don't really have to do with reproduction. So just like musculature and skeletal features, right? Males usually have broader shoulders. Females usually have broader hips, taken as a group typically, right? No offense to anyone, hopefully. And then males usually have a deeper voice. Females usually have a higher voice, right? Males usually have facial hair. Females usually don't have facial hair. So those would be good examples of secondary sexual development. So people with Turner syndrome, because they don't have the proper sex chromosomes, then they typically don't have that primary or secondary sexual development. Because they are missing a sex chromosome, their chromosomes can't form tetrads properly in meiosis, and so they can't form gametes properly, and so they are infertile. For whatever reason, they have a short stature, and then also this is weird. You can't really see it behind the behind my little navigation bar here, but it says webbed neck. So if you go like this, you'll notice that there's kind of like little ligaments right here that kind of stick out, right? So people who have Turner syndrome, they have this just normally, just kind of walking around every day. Like they don't have to do that, and they have those little folds or whatnot. So those are signs and symptoms and a karyotype of someone with Turner syndrome. All right, Klinefelter syndrome. This is what a karyotype looks like for someone with Klinefelter syndrome. You'll notice all our autosomes are correct and good and normal, but then here we have two X's, right, and then one Y. So the way that we would summarize that is 47, because we have an extra chromosome now, right? Normal is 46, right? So 47 XXY, and that tells us right there all that we need to know. That is the reason why we have 47 chromosomes. So again, trying to be sensitive, right? People with Klinefelters have physical appearance of a male. So babysitting, changing a baby who has Klinefelters, changing their diaper, right? Male, obviously. Same thing, right? Symptoms: lack of primary and secondary sexual development. No surprise there. And then same thing, infertility because of this extra chromosome. Then the tetrads kind of get all messed up, and they try to make it like a quadritrad, or I don't know even what it what it would be called. But yeah, so they can't make gametes correctly, and so they can't produce sperm, and so they can't have biologically they can't have their own biological kids. All right, so then trisomy X is the last one that we're going to talk about. So here's what a karyotype looks like for trisomy X. So you'll notice autosomes normal, right? Sex chromosomes, 3X's, no Y. So the way that we would summarize this is 47, because there's an extra one, XXX. That tells us why we have an extra one, so we're good there. Um, so obviously physical appearance of female, because they have more than one X and no Y. So for some reason, unbeknownst to science, they have tall stature, right? There's an increased width between their eyes, so their eyes are kind of a little bit not like really offset, but a little bit offset off to the sides of their heads. Proportionally smaller head sizes, so kind of like cru de chat, right? Microcephaly. Learning disabilities, kind of like Down syndrome, right? Delayed speech and language, which could be a cause of the learning disabilities or could be caused by the learning disabilities. It's nature versus nurture, right? It's that age-old question. Poor motor skills. So motor skills are like fine motor control or fine movement. So if you have really good handwriting, you have really good motor skills, fine motor skills. If it looks like you're writing with your feet, right, then you have poor motor skills, kind of like it looks like my handwriting is sometimes. So people with trisomy X, not all of them have poor motor skills, but some of them, most of them can't do that. All right, so having said that, then I know that you all have questions, and I really wish that we were in class because you guys have amazing questions, and I love the dialogue, and that's part of why I became a teacher, to interact and to teach and to learn myself. But we can't do that because of COVID-19 right now. So maybe I'll maybe preempt some of your questions, and if you have others, you can comment in the comment section, and then I'll get around to that, answering those eventually. So, can we ever have a human born without an X chromosome? Drum roll, please. No. No, never in recorded history, never in science has there been a baby born without an X chromosome. So of course, right? You guys probably knew that, or at least you could guess that, right? The X chromosome carries genes that are essential to survival, right? Remember, X chromosome encyclopedia, Y chromosome comic book, right? Can you deal without a Y chromosome? Yes. Half the people walking around in this world are doing just fine without a Y chromosome. You don't need a comic book, right? Do you need the encyclopedia? Yes, you definitely need the encyclopedia. And then here's one last kind of interesting little tidbit. Is so scientifically, right? All babies, every single one of us, all 8 billion people in the world, start out as female, right? And then if and only if around week 07, there's a gene on the Y chromosome called the SRY gene that around week 7, then that activates and then that starts producing proteins. And once that activates and once those proteins are starting to be produced, then that embryo, that fetus, that baby takes a sharp right hand turn towards what we would call maleness, right? And if that is not present, right, then the baby, the fetus, the embryo would continue on the straight and narrow path to femaleness, right? Which explains why Turner's, which doesn't have a Y but it only has one X, right? They just continue on that straight and narrow path, right? Klinefelter's, even though it has two X's, right, and one Y, that Y is present, and so that Y has that SRY gene on it, and so around week 7, they're going to take the sharp right hand turn, and they're gonna go towards maleness. So then my last slide, if we were in class, I'd give you time to do the karyotype, which is why for our next class period, which depending on which section you're in, is either Thursday or Friday. But our next class period, there's not going to be any new video, there's not going to be any new notes. I'm just going to give you time to work on your karyotype project. Remember, you got to take a picture of that and you got to turn that in either on LMS RenWeb if you can figure out how to do that, or you could email that to me if that's easier for you. And if you are really technologically inclined, you could use like the snipping tool, right, and on a PC in Windows, and you could actually snip out each little individual chromosome and then drag it over to a Word document and kind of do it digitally, and then that would be easier for you to turn in on LMS RenWeb or whatnot. So, anyways, if you have any questions, fire away, and yeah, I will hopefully see you guys soon, maybe a couple weeks after spring break. So, this has been Mr. Leo. Today we learned about non-disjunction and different chromosomal abnormalities. Thank you so much for watching, and I will see you on the flip side.